Hello and welcome back. Through the period of the Renaissance, there was a burgeoning of investigation, which led to scientific method and a way of thinking about the world, not by kind of waving your arms around and guessing or attributing phenomena to supernatural sources, but rather through careful observations, logical thinking, and analysis. A way of seeking trends and drawing generalizations. It's based on assumptions and methods that are reliable, that are objective, and above all, are testable. There are two scientific methods to note, the inductive and the deductive method. The inductive method is such that one makes observations over time and generalizations eventually may be drawn from those observations. It's generally many observations. Anatomy is a product of the inductive method. Scientific observations over time lead to generalizations. An example is the number of ribs in an individual. So by studying skeletons and x-rays, we can see that there are 12 pairs of ribs in the human body. Now you might ask a question, and knowing that, does that attribute to both males and females? Do some people have fewer ribs or more ribs than normal? Well, we do more observations to find these things out. In general, humans have 12 pairs of ribs. Yes, there are exceptions. Males and females are the same. Sometimes individuals may have more than 12 pairs or fewer than 12 pairs. That is an exception to that rule. The deductive method, on the other hand, requires experimentation. These observations that we have may lead to questions. So for example, we can observe that during birth there are uterine contractions. What causes those uterine contractions? Well, once we have a question, we form a hypothesis. In this case, there may be a number of hormones and chemicals responsible for uterine contractions. Let's say we choose one, oxytocin. Oxytocin causes uterine contractions. Well, can we test that hypothesis? If we can't test it, then we can't make it. But we can test this particular hypothesis, which I did in university with frogs. And what you do is you isolate the muscle of the uterus, and you put the muscle, we used frogs, we used the muscle in saline solution. We had a control that had no oxytocin, and we had a treatment that did have oxytocin. Of course, you can't just do it once with one muscle. You need to do it several times and have replicates in your experiment. But you always need a control. So we can isolate the muscle tissue, we can apply oxytocin, and we use a control where oxytocin isn't used, but all other variables are as similar as we can make them. Things like temperature should be similar, of course, pH, the acidity of the solution, the saline content. The only difference is oxytocin. That's the treatment. And through experimentation, we find that yes, oxytocin does indeed cause uterine contractions. Our experimental design to find this out must include a sufficient sample size, a large sample size, because with a small sample size, for example, if we're looking at plants and we're testing a fertilizer, if we have a very small sample size, it could be that all those plants are subject to a pest invasion. So we need to have a large sample size. We need a control group separate from the treatment group because we need to compare our treatment with something so that we know the reaction that we got was due to the treatment and not due to some other factor. We must prevent psychosomatic effects. In that case, we use a placebo. If we're doing tests on drugs, for example, and we have a clinical trial of humans, we use a placebo, which is in shape and size and probably flavor the same as the treatment, but it is not. It doesn't have any effect at all. I think in the old days they used like sugar cubes. So we can use placebos. We can avoid experiment bias by not informing the person administering the treatment or the placebo of which they are administering, because body language is something that gives us away. So somebody who's administering the placebo might be going, maybe not that obvious, but we're so good at reading body language that we could tell in some cases. And that's called a double-blind study that avoids experimenter bias. Also, we do statistical testing, and that ensures that the difference between the treatment group and the control group was not random. Sometimes we might see a difference, a small difference that's just due to chance alone. But we need to know that it was due to the variable being tested. Indeed, in medicine, we need to be 99% sure that the result was due to the variable being tested. Another check or balance to our science methods is peer review. When you do an experiment and you write it up in a paper, you submit it to a scientific journal, then your paper is sent to other scientists in the same field. So if you're a cardiologist, for example, you're doing investigations into cardiac experimentation, then your paper will be sent to other cardiologists for review, and they will inspect your methods. This ensures honesty, objectivity, and quality in science. So as professionals, you and I, we need to dispel myths about health. In order to do that, we need to be able to read these scientific journals. And in order to do that, we need to come to understand the terminology around anatomy and physiology, which is what this course will do. Now in science, we need to distinguish between facts, laws, and theories. A scientific fact is information that can be independently verified by a trained person. For example, iron deficiency, a trace element, leads to anemia. That can be verified by a scientist. A law is a description of the way matter and energy behave. It results from inductive reasoning, repeated observations over time. It's generally written as statements or mathematical formulae, such as, for example, the first law of thermodynamics, which is that energy can not be created or destroyed, but it changes. Theory, the word theory is another story. A person who is not a scientist or not into science may think a theory is the same as an hypothesis. But in science, a theory is not a hypothesis. An hypothesis is an educated guess that must be tested. A theory, rather, is a summary of conclusions drawn from observable facts. So this may be hundreds, even thousands of experiments and observations. It provides explanations and predictions, such as the sliding filament theory of muscle contraction, where actin and myosin are responsible for cells contracting. Also, the theory of evolution by natural selection. That has been an observable and a testable theory over many years and hundreds of experiments, even thousands. So there we come to the end of the scientific method. I'd like to stop there. Thank you very much for watching.